body fat and look at that situation. So there are several things that could cause fat to be broken down. These are two big signals telling adipose tissue to start mobilizing their energy reserves: adrenaline or epinephrine and glucagon. Remember, glucagon is the flip side of the insulin story. So insulin tells tissues to absorb glucose out of the bloodstream, tells the liver to store it as glycogen. Glucagon does the opposite. It tells the liver to release glucose into the bloodstream, to break down the stored polysaccharide and release it as glucose. At the same time as doing that, it's going to fat cells and telling them, okay, start breaking down some stored fat. Because this appears when blood glucose is low. When you don't have much blood glucose, your total body energy reserves or energy availability is low. So it's mobilizing not only glucose in the liver, but fatty acids in the fat cell. And it comes knocking at the door there. It binds to a receptor of the fat cells, and that leads to a second messenger inside, cyclic AMP, which turns on the breakdown pathway for fat mobilization through phosphorylation. This picture here probably tells the story better. So this cell on the left represents a white fat cell, an adipose cell. Here's a capillary that we'll find in the smallest of the blood vessels. Blood vessels and serum flowing through here. Quite conveniently, on the other side, we put a heart muscle cell, just to compress everything into one. So here comes, in the bloodstream, something like adrenaline or glucagon, and it sees this receptor. You have biological recognition going on there in the usual way. That docks there with a special receptor binding site. When it rings that doorbell by binding, it stimulates conformational changes in the receptor, goes through a complex chain of events we won't really get into, but this is one highlight of that chain. This is very typical of a lot of hormones. The docking of a hormone at the receptor often leads to the conversion of ATP into cyclic AMP. This is referred to as the second messenger, where the primary messenger is the hormone floating around in the bloodstream. It stays on the outside, it's not internalized, but it's binding there, stimulates the production of this internal second messenger, cyclic AMP, whose job it is, you've got lots of cyclic AMP being generated, floats around through the cell, activating this enzyme through allosteric activation. This enzyme is a protein kinase A. Remember, there's two general strategies for activating pre-existing hormones or pre-existing enzymes. You can use an allosteric mechanism, like this allosteric that stimulates this enzyme. The second way is to put on a post-translational covalent modification. Well, with hormones, this is a typical story that cyclic AMP leads to the activation of a kinase, which puts phosphates on target molecules inside the cell. One of them is the lipase. It's called hormone-sensitive lipase. In the presence of cyclic AMP, this enzyme puts a phosphate on the lipase, which activates it. So this lipase, which breaks down the stored lipids, is there already, but it's not active in most cases. It has to be hormonally activated through this hormone system. So you